Okay, so welcome to this next video in which we are discussing the cell cycle. Okay, so we're currently discussing the S phase. So the origins of replication have now fired, and you are in the process of copying the DNA. Now you want to make sure that you can't reassemble pre-replicative complexes on our two new origins of replication. Okay, so the instant you produce this second new origin of replication, a new origin recognition complex will bind there, but you want to make sure that you're not going to get CDC6 binding and CDT1 binding, because you don't want the MCM ring to bind, and then you don't want it to form a pre-initiation complex. So what the cyclin A CDK2 complexes do is they phosphorylate the origin recognition complexes, which stops them from associating with CDC6. In addition, they phosphorylate CDC6 itself, and this targets it to be destroyed, so the levels of CD6 go right down within the cell. Meanwhile, CDT1 cannot bind to the origin recognition complex anymore anyway, because CDC6 was needed to bind before CDT1 could bind. However, it also gets another protein bound to it to sequester it inactive until another round of the cell cycle. Okay, and this protein that binds to it is known as geminin. Okay, so I'll colour geminin here in blue. So after the CDT1 has done its job, geminin will bind to the CDT1. Okay, now where does the geminin come from? Well, geminin has its levels upregulated by E2F transcription factors. So remember, E2F transcription factors took us into this S phase of the cell cycle. They increased the expression of cyclin E, cyclin A, and now we've also seen they increased the expression of geminin here. Okay, by increasing the expression of cyclin E, we form cyclin E CDK2 complexes, which cause the firing of the origins of replication. By increasing the expression of cyclin A, we form cyclin A CDK2 complexes, which are involved in trying to prevent new pre-replicative complexes being formed. Okay, and we also produce geminin, which also helps in this job of preventing double replication of the chromosomes effectively. Okay, right, so where do we stand then now? What have we done? Well, we have now replicated the DNA. So we now have a large cell, okay, so it's been through the first growth phase, and therefore it's nice and big now. And we have 2 copies of every chromosome within the nucleus now. Okay, so this now isn't representing the double strands of DNA. Each one of these lines is representing a chromosome. Okay, so we have 2 identical chromosomes here, both in red. So this isn't representing double-stranded DNA, I'll repeat it again. So this isn't one strand of DNA and this isn't the other. These are the 2 chromosomes, basically. So each of these lines is now a piece of double-stranded DNA. Okay, and these 2 utterly identical chromosomes are now called sister chromatids. Okay, right. And I should just mention that the sister chromatids are kept attached to one another. So the 2 pieces of identical DNA, those are kept attached to each other. And the way they are kept attached to each other is by protein complexes known as cohesins. Okay, so let me just discuss with you what a cohesin is then. Okay, well basically a cohesin is a complex consisting of 4 separate proteins that wraps around the 2 strands of DNA. Okay, so if we've got our 2 chromosomes here, here's one and here's the other. Okay, so both of these lines represent double-stranded pieces of DNA. Okay, now of course the double-stranded pieces of DNA will be wrapped around histones, but we won't bother drawing all of that. The cohesins are going to wrap around the 2 pieces of DNA and hold them together. So I'd just like to discuss the 4 proteins that are involved in producing a cohesin complex. Okay, right. So we're going to start off with 2 proteins that have a very similar structure, which are known as SMC proteins. Now, what does this stand for? This stands for structural maintenance of chromosomes. Okay, so the S is for structural, the M is for maintenance, okay, and the C is for of chromosomes. Right, so there are going to be two proteins which are in this class of structural maintenance of chromosomes proteins. Okay, so let me discuss what the structure of these proteins is. They have what is known as a coiled coil structure within them. Okay, so what does this mean? Well, basically, if we start off with the amino terminus here, okay, before I describe to you what this ball is, I want to first describe what the coil is. So these structural maintenance of chromosomes proteins, they have a number of interesting domains. I'm going to start off with the coiled coil domain. So initially, ignore this portion here. In fact, actually, I'll tell you what a coil domain is before I tell you about the structure of a structural maintenance of chromosomes, because a coiled coil domain is something that you have in far more proteins than just the structural maintenance of chromosomes proteins. Okay, so firstly, to discuss what coiled coil domains are, we firstly need to discuss what an alpha helix is. So basically, let this line represent the polypeptide, okay, so the polymer of amino acids. What can happen is it can fold up into an alpha helical structure, which is basically a spring-like structure, okay, like so. So this here, this is the concept of an alpha helix, and I'll colour this in blue here. Okay, so alpha helices are a type of secondary protein structure that is held together by hydrogen bonds that form between the peptide links on one strand, let's say this one here, and the peptide links on a strand below it, okay? So the portion below it, it will form hydrogen bonds between their peptide links, basically, and this holds the whole thing in this spring structure here, known as an alpha helix. Okay, so an alpha helix is just the fancy term for a spring structure, effectively. Okay, so what's a coiled coil domain? Well, basically, a coiled coil domain is going to be made up of a really long alpha helix. Okay, so now when I draw a single line, this is now going to represent an alpha helix. Okay, and what's going to happen is the alpha helical polypeptide is going to fold up into a larger structure, basically. Okay, where it coils around itself. Okay, like so, and then it will fold back and overlap with itself to form like this double helix, effectively. Okay, so this is what a coil domain is. Each one of this line here, this no longer represents a polypeptide. This represents a polypeptide that is in an alpha helix. So if I wanted, I could draw it more like this. Okay, in fact, this probably gets the message across better. So the whole thing within the alpha helix is now coiling up into a double helix. And what's it helixing with? What's helixing with itself, okay? So the full thing is now going to turn around, and then it's going to intertwine with itself like so. Okay, and such a domain which has this alpha helix coiled around itself like this, this is known as a coiled coil domain. Okay, so this full thing is a coiled coil domain. So, these structural maintenance of chromosome proteins, these are going to contain something along the lines of this. So, the other important domains that these structural maintenance of chromosome proteins have is they have two ATPase domains, and they have a portion down here, effectively, which is the linker between the two alpha helices, which are going to coil around each other in this way. Okay, and that's known as the hinge region. So let me show you this then. So we'll have this hinge region down here, and then we'll have the alpha helices which coil around each other like so. And now I'll show the alpha helices just as a line because it's easier to draw. Okay, and these alpha helices are coiling around each other like so. And then right at the ends over here, you have, here's the amino terminus, let's say, and here's the carboxylic acid terminus. And these two special domains that I'm showing as little balls here, these are ATPases, okay? So they have the enzymic capacity to hydrolyze ATP. So I'll colour the ATPases at the ends in turquoise. Then we'll colour the coiled coil domain which is made up of these two alpha helices. Here's one in vivid purple there, and here's the second, which I'll colour in green here. Okay, and then connecting the two alpha helices, you then have this structure in the middle, which is blue, and this is known as the hinge region. Okay, so in blue here, this is the hinge, and then this is the coiled coil domain here, made up of these two coiling alpha helices. Okay, so coiled coil domain. Okay, right. So you're going to have two of these structural maintenance of chromosomes proteins. One of them is going to be a structural maintenance of chromosome protein 3, and the other is going to be a structural maintenance of chromosomes protein 1. Okay, so let's say this one here is a structural maintenance of chromosomes protein 3, an SMC3 protein, and it's going to bind with another SMC protein. So it will bind at the hinge region with another SMC protein, and this one is going to be the SMC1 protein. Okay, so once again I'll draw the coiled coil structure here. Okay, like so, and then you'll have the other alpha helix that makes up the other side of the coiled coil. Okay, like so, and then here are the two ATPase domains again. So I'll colour the ATPase domains in turquoise once again, and then I'll colour the two alpha helices which make up the coil domain again in those same colours. So here in purple is one of those alpha helices, and then in green we'll have the second of those two alpha helices. Okay, like so. Right, so this protein on the bottom then, this is SMC1, the structural maintenance of chromosomes protein 1. And now, at this other end, you're going to have additional proteins binding here, okay, so to the ATPase domain. So this should be in contact with these ATPase domains ideally. Okay, so these will be bound here, and this will also be bound here. Okay, and this protein here is what's known as SCC1. Okay, and I'll colour SCC1 in yellow here. And then finally, on top of SCC1, you have another protein bound, which is SCC3. Okay, so on top of this, you've also got SCC3, which I'll colour in red. So this complex of four proteins now, the SMC3, the SMC1, the SCC1, and the SCC3, this is called a cohesin. Okay, and you can see that it forms a ring, basically. And this ring is going to have the two strands of the DNA running through it. So effectively, what you're going to have is the cohesin wrapped around like so. Okay, so if these are the hinge regions here, okay, which I'll colour in blue. Okay, so these are the hinge regions of the SMC3 and SMC1. Then we have the coiled coil domain here in purple, going round into the ATPase domain here, and then again we'll have the coil domain here going round into the ATPase domains. Okay, and I'll colour these in turquoise. And as you can see, one is going behind the two strands of the DNA, and the other is coming out in front of them. And then bound over here, finally, you then have the two SCCC proteins, SCC1 here, okay, and then SCC3 as well here. Okay, and that makes up our full cohesin, and basically all the way along the two sister chromatids you will have these cohesin complexes which are holding the two sister chromatids together. Okay, right, so that's what's holding these two sister chromatids together, and the knowledge of the cohesins will become important later when we finally get into M phase. Okay, so S phase is now complete, and we're going to go into the next phase of the cell cycle, which is G2 phase. Okay, now in G2 phase, what's going to happen is the cell is going to further grow. Okay, so you can either view this as the second gap phase or the second growth phase. You're going to continue growing the cell. You're going to be producing more proteins. You're going to be duplicating more intracellular organelles. Okay, in addition, you're going to be getting ready to move into N phase. Okay, now how do you go from G2 phase to N phase? Because there is a very rigorous checkpoint that you have to go through. Okay, so there is a hurdle you have to get over to move from G2 to N phase. So just like there was that control switch to move from G1 to S, there is a control mechanism to move from G2 to N, and we now want to discuss what this control mechanism is. Okay, so basically throughout the second growth phase, what's going to be happening is you're going to be producing another cyclin. Okay, and the cyclin that you start producing in the second growth phase is cyclin B. Okay, so another one of these cyclins that's going to go up during a certain portion of the cell cycle. Okay, so cyclin B is going to go up in G2 phase. Okay, and it's going to get higher and higher and higher and higher. Now, cyclin B once again is going to bind to a cyclin-dependent kinase. So let's see the cyclin-dependent kinase it's going to bind to. Okay, so we're going to meet another cyclin-dependent kinase, and this cyclin-dependent kinase is cyclin-dependent kinase 1, the archetypal cyclin-dependent kinase. Okay, right. So we'll colour cyclin-dependent kinase 1 in orange here. What's now going to happen then is throughout G2 phase, when you're producing the cyclin B, you're going to be getting cyclin B binding to cyclin-dependent kinase 1. Okay, so here is the cyclin B binding to the cyclin-dependent kinase 1 enzymes here. Okay, and they're forming another cyclin-dependent kinase complex here. Okay, so this complex is called a cyclin B CDK1 complex. But it's also got another name, just like all the other ones had other names. This complex of cyclin B with CDK1 is also called the M CDK, because it's going to be extremely important in producing the mitotic phase of the cell cycle. Cool. Okay, now, so throughout G2 you are accumulating more and more cyclin B, which is binding to cyclin-dependent kinase 1. And you might say, hang on a moment, if this is the M CDK, then surely it should only become active in the mitotic phase, and indeed it does. CDK1 is going to bind the cyclin B within the G2 phase, but it's not going to be activated yet, and I want to now explain the reason it's not going to be activated. So, cyclin B CDK1 complexes are going to be phosphorylated after they form. So after the cyclin B binds to the CDK1, what's going to happen is they're going to be phosphorylated by two opposing forces, basically. One phosphorylation is going to be performed by an enzyme known as the cyclin-dependent kinase activating kinase. Okay, activating kinase. And for short, the cyclin-dependent kinase activating kinase is often abbreviated to the CAK. C for cyclin-dependent kinase, A for activating, and then K for kinase. Okay, so I'll draw this here. So one of the forces that's going to be acting on the cyclin B CDK1 complexes is this cyclin-dependent kinase activating kinase, which is going to phosphorylate CDK1 on a threonine residue. Okay, so here it comes. It's going to phosphorylate a threonine residue on cyclin-dependent kinase 1. I even have the exact number of the threonine residue that it's going to phosphorylate. It's going to phosphorylate threonine 161. Okay, and this activates CDK1 when it's bound to cyclin B. So this is going to activate the cyclin B CDK1 complexes. And you might say, aha, so this is what's going to turn them on. Okay, when we go into M phase, this enzyme is going to become active and it's going to turn them on. Well, no is the answer. It gets more complicated. The cyclin-dependent kinase activating kinase will be phosphorylating cyclin B CDK1 complexes even in the G2 phase of the cell cycle. Okay, you might say, but that will make it active, surely. Well, no, because there's also going to be another opposing force. Okay, so there's also going to be another kinase enzyme over here. Okay, and this other kinase enzyme is going to be called WEE1. Okay, and I'll colour in WEE1 in green. Now, this is also a kinase enzyme, and this is another one of these kinase enzymes that can phosphorylate tyrosine residues as well as threonine residues, so it's an unusual one. Okay, and it's going to phosphorylate two residues that are right in the active site of the cyclin-dependent kinase 1. Okay, so there is a tyrosine residue within the active site, and there is a threonine residue. So the single letter amino acid code for tyrosine is Y. Okay, so Y stands for tyrosine. Tyrosine could not have T because T had already gone to threonine. Okay, so threonine got the T. Okay, so you have a tyrosine residue and a threonine residue within the active site of your cyclin B CDK1 complex. Okay, specifically the tyrosine is number 15 and the threonine is number 14. Okay, so these two residues are going to be the target of this WEE1 kinase enzyme. So it's going to phosphorylate this tyrosine residue, and it's also going to phosphorylate this threonine residue. Okay, and when the cyclin B CDK1 complex has these residues within its active site phosphorylated, this is going to inhibit the cyclin B CDK1 complex. So it's not functional. So, to summarize then, in the G2 phase of the cell cycle, not only are you further growing the cell, okay, you are also preparing to go into the M phase, and for this preparation, what you are doing is producing more and more cyclin B. This cyclin B is binding to CDK1 to form cyclin B CDK1 complexes, and the instant these cyclin B CDK1 complexes are formed, they get phosphorylated by both the CDK activating kinase, called CAC, okay, so this is CAC here, which will phosphorylate them on threonine 161 and activate them. But they're also getting phosphorylated at tyrosine 15 and threonine 14 by WEE1 kinase enzymes, which will inhibit the cyclin B CDK1 complex. So at the moment, these cyclin B CDK1 complexes that we're forming more and more of, they are not going to be activated. Okay, they're going to be sitting within the cytoplasm of the cell, not doing anything. We're just accumulating more and more of them. And to move from G2 to M, what we're going to have to do is we're going to have to remove these phosphate groups that are on the tyrosine 15 and the threonine 14. Okay, and once we've removed them, now the thing will become activated. So at the G2 to M transition, what we're going to do is unleash the cyclin B CDK1 complexes by removing these phosphate groups on these residues that are within the active site. Okay, and we'll see how that's done in the next video.